Imagine standing outside on a warm summer night. The air is thick and humid, maybe after a rain shower,
Yeah.
and all around you, in the grass and the trees, tiny points of light blink on and off. Fireflies.
Mm, that classic image.
It feels like pure, unadulterated magic, doesn't it? That ephemeral glow.
It really does.
But what if that wasn't just a beautiful, transient light show? What if that ability to create light from within held the key to unlocking incredible scientific potential?
Like tracking pollution, maybe.
Exactly, or diagnosing diseases at a molecular level, or even illuminating our world in completely new, sustainable ways.
That's the fascinating intersection we find ourselves in today. Bioluminescence is ancient, a fundamental part of life's evolutionary toolkit,
Right.
but it's also at the forefront of modern biotechnology and scientific discovery.
So it's not just fireflies and weird deep-sea creatures.
Oh, far more than that. It's
Yep.
far more than just the twinkling lights we might see on land or the, eerie glows from deep sea.
And that's exactly what we're diving into today. A deep dive into the luminous world of bioluminescence.
Okay.
We've pulled together a stack of sources for this exploration, everything from detailed research papers mapping the intricate chemistry and biology
Yeah, the nitty-gritty stuff.
to broader articles exploring the sheer diversity of organisms that glow, and cutting-edge notes on how this natural phenomenon is being engineered and harnessed for human applications.
Right. So a real mix of the fundamental science and the applied side, too.
Our goal in this deep dive is to navigate that sea of information with you. We want to cut through the technical jargon and the volume of data,
Yeah, make it accessible.
to distill the most important insights, uncover the truly surprising facts, and give you a clear understanding of why this natural glow is not just aesthetically beautiful, but also profoundly useful and scientifically powerful.
Think of it as a shortcut, maybe. A shortcut to being well-informed on a truly luminous topic.
Okay, let's unpack this. To understand the applications, we have to start with the source. What exactly is bioluminescence at its most basic level? What makes a living thing glow?
At its core, it's a chemical reaction. Simple as that, really. Inside a living organism, a reaction produces light.
Not like fire, though.
Right. Unlike a fire, which produces light by burning and generating a lot of heat, bioluminescence is often called cold light.
Cold light.
Yeah. Very little energy is lost as heat. Most of it is converted directly into light photons. It's remarkably efficient in that sense.
And the sources break down the key components involved in this reaction. It's not just one thing glowing.
That's right. You need a couple of main players, typically. For the light-producing reaction to occur, you usually need two main types of molecules.
Okay.
First, there's the luciferin. This is the molecule that actually gets oxidized and, in the process, emits the light. Think of it as the fuel or the emitter.
The fuel, got it. And the
Yeah.
second
Second, you need an enzyme called a luciferase. This enzyme acts as the catalyst. It speeds up or enables the specific chemical reaction that makes the luciferin glow.
So the luciferase is like the switch or the engine that makes the luciferin fuel burn just right to produce light.
That's a good way to think about it, yeah.
Yeah.
And importantly, for most, though not all, bioluminescent systems, you also need oxygen.
Ah, oxygen.
Oxygen is often the oxidizer, the molecule that reacts with the luciferin in the presence of the luciferase to generate that light.
And our sources highlight the firefly system, specifically firefly luciferase or fluke and its partner D-luciferin, as a fundamental example and a key tool. Why is that one so prominent, especially in the lab?
Well, the firefly system is incredibly well studied, partly because it's terrestrial and relatively easy to access compared to, say, deep-sea organisms.
Right, easier to catch a firefly than an anglerfish.
Pretty much. And partly because it's remarkably efficient.
Yeah.
D-luciferin is the specific chemical molecule, and fluke is the enzyme. This particular pair is very widely used in laboratories as an in vitro reporter,
An in vitro reporter. Meaning what exactly?
meaning scientists can introduce the components of the firefly system into cell cultures or test tubes.
Go.
When the conditions are right, when the biological event they're interested in happens, the fluke and D-luciferin react and produce light.
Ah, so the light is the signal.
Exactly. The presence and intensity of the light then tells the scientist that the event occurred. It's like a molecular indicator light they can use to track processes inside cells or biochemical reactions.
So it's a simple chemical reaction in a living organism that creates light. That sounds simple.
Hey, well, the basics are fairly straightforward chemistry.
But the sheer diversity of how life uses this trick in nature is anything but simple.
It's absolutely stunning. The sources make a point of emphasizing that bioluminescence is not a single invention that life stumbled upon and spread. It has evolved independently multiple times across the tree of life.
Multiple times, really?
Yeah, we're talking at least 40 separate origins, maybe more we haven't found yet. And get this, within just one group, like the ray-finned fishes, it's popped up 27 distinct times.
27 times in fish alone. That tells you this isn't just a neat trick. It must be a seriously powerful evolutionary advantage in diverse environments.
Exactly. It confers a real benefit. And the ways organisms use it are incredibly varied and often surprising. It's a whole toolbox.
A toolbox of light. Okay,
Yeah.
what's in the toolbox?
Let's start with a use that seems counterintuitive. Using light to hide. How does that work?
Hiding with light. That feels backwards. How can light help you hide?
It does, doesn't it? But in the open ocean, particularly in the mesopelagic or twilight zone where faint sunlight filters down from above.
Okay, that dim, deepish part of the ocean.
Right. Many animals there use a technique called counter-illumination. They have light-producing organs called photophores, usually on their undersides.
Photophores, light organs.
Yep. And they precisely control the intensity and color of the light emitted from these organs to match the dim, downwelling light from the surface.
Ah, so if a predator is looking up from below, instead of seeing a dark silhouette against the faint light, they see nothing.
Precisely. The animal's underside blends into the background light, effectively making them invisible from below. Organisms like hatchetfish, many squid species, and numerous other midwater fishes are masters of this.
Wow.
It's a sophisticated form of active camouflage. Pretty clever.
Okay, so light for camouflage, that's amazing. What about the more intuitive uses, like attraction?
That's probably the most familiar role. Fireflies are the classic example, right?
Yeah, we all know them.
Their species-specific flash patterns aren't just random blinks. They're complex signals for finding mates.
So it's like they're talking to each other with light.
Exactly. Males fly around, emitting a particular sequence or timing of flashes, and receptive females, often perched on vegetation, watch for the correct signal and flash back. It's a luminous conversation.
A date signaled by flashes.
Cool. And some click beetles also use an orange glow when flying, likely as an attractant too.
They're quite literally putting out a come-hither signal with light.
Exactly. And it's not just for romance. Light is a potent lure for prey.
Ah, the anglerfish. I was waiting for that one.
The anglerfish, with its iconic dangling lure, is a prime example. That lure, the esca, contains bioluminescent bacteria living symbiotically within the fish.
Bacteria inside the lure.
Yep. The anglerfish can control the blood flow and muscle contractions around the esca to make the light flicker or move, mimicking a smaller organism.
Tricking its prey.
Totally. Unsuspecting prey are drawn in by the apparent meal and find themselves right next to a very large, toothy mouth. Dragonfish use similar glowing lures as well.
Symbiosis again. The bacteria get a protected home and nutrients, and the fish gets living date.
It's a classic win-win in the deep sea. And this symbiotic relationship with luminous bacteria is incredibly widespread. Our sources mention that these bacteria are ubiquitous in marine environments.
Everywhere in the ocean.
Pretty much. Found in seawater, sediments, the guts of marine animals, and they rapidly colonize dead or dying organic matter.
Which explains that historical observation of dead fish or meat sometimes glowing in the dark.
Exactly. That glow often comes from these symbiotic or saprophytic luminous bacteria colonizing the tissues.
Yeah.
The sources also note that while these bacteria are generally not pathogenic to humans, basic safety precautions are always wise if you're culturing them in a lab or at home.
Good to know. And this bacterial glow can even help with dispersal, right? Like for fungi.
Yes, that's another fascinating, less intuitive use. Some terrestrial fungi, belonging to genera like Armillaria, Mycena, and Pinellus,
Glowing mushrooms.
or glowing mycelium, the sort of root-like network, they produce a persistent, often greenish glow, usually from their mycelium in rotting wood, or sometimes from their fruiting bodies, the mushroom part.
Why would they do that?
One leading hypothesis is that this light attracts night-flying insects,
Okay.
which then pick up and disperse the fungal spores, helping the fungus spread to new locations.
Like little fungal taxis attracted by the light.
Kind of. There's also the example of luminous bacteria infecting nematodes that parasitize caterpillars. The glowing caterpillar carcass might attract predators or scavengers, aiding the dispersal of the bacteria and the nematodes they carry.
Wow, intricate relationships. So light can attract mates, prey, or even help spread spores and symbionts. What about offense? Can light help an organism ward something off?
Absolutely. Just like bright colors in the daytime can signal danger or toxicity, think poison dart frogs.
Right, don't touch the bright frog.
A glow at night can serve as a warning, a form of aposematism. Firefly larvae, for instance, are famously unpalatable due to defensive chemicals, and their persistent glow serves as a clear signal to predators. Don't eat me.
So the glow means I taste bad.
Pretty much. Some millipedes use a similar strategy. In the marine world, organisms like certain scale worms, jellyfish, and brittle stars may also use light as a defensive warning, though the precise function can be complex and is still being studied.
There's also that unique freshwater snail, right?
Ah, yes. The sources mention Latia neratoids, which is unique as the only known freshwater mollusk that's bioluminescent.
What does it do?
It produces a glowing mucus when disturbed, which is thought to deter predators.
Glowing slime.
Yep. And a marine snail, Hinia brasiliana, uses its shell not just as protection, but as a diffuser, flashing bright light through it, also believed to be a defensive signal.
A snail using its shell like a lampshade. Nature is truly inventive.
It really is. Another defensive tactic is confusion. Some squid, when startled by a predator, don't release a cloud of dark ink like their relatives.
No ink. What then?
Instead, they squirt out a burst of bioluminescent liquid or particles. This creates a sudden, dazzling flash of light that can disorient or distracts the predator.
Like a flashbang?
Sort of, giving the squid precious seconds to escape. A luminous smokescreen, basically.
A luminous smokescreen. That's brilliant.
Exactly. Beyond obvious attraction and defense, light is also used for communication in ways we don't fully understand yet. We mentioned fireflies for mating. But the sources also point to Quantula striata, the only known terrestrial mollusk, a slug, that emits light pulses, and its communicative function isn't completely clear. We're still figuring that one out.
Still mysteries out there. And finally, the sources talk about mimicry. Can organisms use light to impersonate something else?
Yes, both offensively and defensively. Aggressive mimicry, as we discussed, is seen in anglerfish and dragonfish using their lures to mimic prey signals.
The fake bait trick.
Right. But a particularly clever example is the cookie cutter shark.
Cookie cutter shark. Sounds cute.
Not so much. These small parasitic sharks have photophores on their undersides for counter-illumination, blending their silhouette against the downwelling light.
Okay, like the hatchetfish.
Yes, but here's the twist. They have a dark patch among these photophores. When viewed from below against the faint light, this dark patch and the surrounding counter-illumination creates a silhouette that looks exactly like a small fish.
So it makes itself look like a tasty snack to attract bigger fish.
Precisely. A larger predator sees this convincing small fish silhouette swimming along, comes closer to investigate or grab an easy meal.
And then...
And the cookie cutter shark then bites a neat plug right out of the much larger animal's flesh. It's using mimicry to turn predator into prey, or at least into lunch.
That is horrifyingly brilliant. Mimicking a snack to become a parasite on a giant.
A very effective strategy for the shark. The sources also mentioned a debated case of potential defensive mimicry.
Oh, yeah.
The claim that Luciferin cockroaches might mimic toxic click beetles by glowing. However, the sources are careful to note that conclusive evidence for this claim, including whether the cockroaches are truly bioluminescent themselves or using symbiotic bacteria, is still lacking and heavily debated.
Ah, okay. So that one's still up in the air.
Very much so. Science requires solid proof.
It's good to know science is keeping the bar high for evidence. Is light ever used simply to illuminate? Like a flashlight?
It's rare, actually.
Yeah.
Especially compared to attraction or defense. Most marine bioluminescence is blue or green.
Because those colors travel best in water. Right?
Yeah, exactly. Because those wavelengths travel furthest in water. But a few deep-sea barbelled dragonfish, like Aristostomias and Malacosteus, have a remarkable adaptation, a photophore beneath their eye that emits red light.
Red light in the deep sea. But you said red light gets absorbed quickly by water. Doesn't that defeat the purpose?
Exactly. That's the trick. Red light essentially doesn't exist in the deep sea unless you make it yourself. Many deep-sea creatures are red because red appears black in the dominant blue-green light, providing camouflage.
Right.
These dragonfish can shine a red spotlight onto their surroundings, making red pigmented prey clearly visible to them, while the prey remains invisible to most other predators that can only see blue or green light.
It's a stealth vision system, like biological night vision goggles, but red.
Pretty much. They might also use this exclusive red light to communicate with each other without being seen or heard by other species. A secret channel.
So from sophisticated camouflage and cunning lures to defensive warnings and secret flashlights, Bioluminescence is this incredibly versatile, independently evolved trait.
Absolutely. A whole suite of tools.
But this natural magic show isn't just confined to ecological roles, it's become a powerful tool for human science and technology.
That's where the biotechnology aspect comes in. People recognize the potential of natural light for practical uses surprisingly early, long before modern molecular biology.
How early?
The sources mention a fascinating historical note: dried fish skins, which would glow faintly due to colonizing bioluminescent bacteria.
The same bacteria we talked about.
Yep. Those were sometimes used as a weak light source in coal mines in the past.
In coal mines, before electric lights or even safety lamps.
Yes, using candles or open flames in coal mines was incredibly dangerous due to explosive methane gas.
Right, explosions.
A cool biological light source, however weak, avoided that risk. A very low-tech but ingenious early application.
Wow. All right, fast forward to today, and bioluminescence is absolutely central to a lot of scientific research. We already touched on the firefly, flute, and luciferin system being a key lab tool. What makes it so valuable?
Besides its accessibility compared to, say, a deep-sea system, the Firefly system is known for its high quantum yield.
Quantum yield. Unpack that for us simply. What does that mean?
Think of it as efficiency. How much of the chemical energy put into the reaction comes out as light energy.
Okay, light out versus energy in.
Exactly.
Hmm.
The sources state the Firefly luciferase with luciferin can achieve a maximum quantum yield of 41% at optimal conditions, around pH 8.5.
41%? Is that good?
That's remarkably high for a biological system. It means it's very efficient at converting chemical fuel into visible light photons, much better than, say, an old incandescent light bulb.
Right, those wasted a lot of heat.
Precisely. And the Firefly system has a unique feature that makes it particularly useful. Its reaction is directly dependent on ATP.
ATP, adenosine triphosphate. The cell's energy money.
That's the one. The main energy currency of living cells. Luciferin requires activation by ATP before flute can make it glow. This is different from many other bioluminescent systems, like those using covalentrazine.
And how does that ATP dependence translate into a lab tool?
Because the light output is directly proportional to the amount of ATP present.
More ATP, more light.
Yep. You can use the floaty luciferin system as a highly sensitive way to measure ATP concentration in biological samples.
Why would you want to measure ATP? What does that tell you?
ATP levels are a direct indicator of the metabolic activity and viability of cells. If a cell is alive and healthy, it has ATP.
And if it's dying?
If it's dying or stressed, ATP levels drop. This has incredibly broad applications. In basic research, it helps understand cellular energy metabolism. In industry, it's a cornerstone of rapid hygiene monitoring.
Hygiene monitoring, like cleaning checks in factories or hospitals.
Exactly. Companies in food processing, like fish or milk production, or hospitals, can use ATP-based bioluminescence assays to quickly check surfaces for contamination.
How does that work?
If they swab a surface and detect significant ATP, it means there's biological material there, bacteria, yeast, or even human cells from touch, indicating inadequate cleaning.
Because any living cell, or even recently dead ones, will have some ATP.
Right, it's a very sensitive method, capable of detecting ATP levels down to attomoles.
Attomoles, that sounds tiny.
It is, roughly equivalent to the amount of ATP in a single bacterial cell, using commercially available kits and sensitive instruments called luminometers. It's also used in research to monitor reactions that produce or consume ATP.
So incredibly useful for both fundamental understanding and practical application. But the sources also mention limitations to this Firefly system, right? It's not perfect for everything.
No system is perfect, no. Natural systems evolve for specific ecological niches, not necessarily for laboratory convenience.
So what are the downsides?
One limitation is that the reaction products like dihydroluciferyladenylide and oxyluciferin can actually inhibit the enzyme and reduce the total light output.
So it sort of gums up its own works.
A bit, yeah. It can limit the effective quantum yield in some conditions. Also, the light emitted peaks around 558 nanometers, which is in the yellow-green part of the spectrum.
And you mentioned that for imaging deep inside living things, you want redder light, like really red.
Exactly. Light in the blue and green range, and even yellow, is strongly absorbed by components in animal tissues like hemoglobin in blood and melanin in skin.
So it doesn't get through very well.
Right. For in vivo imaging, seeing what's happening inside a living animal like a mouse, you ideally want light in the far red or near infrared range, above 600 or even 700 nanometers, because it penetrates tissue much better. less scattering, less absorption.
Okay, so firefly luciferin is great, high quantum yield, ATP dependent, but maybe not the best color for seeing deep inside a mouse. What are the alternative systems the sources discuss?
Another major class of systems, widely used in the lab, are based on coentyrazine as the luciferin molecule.
Coentyrazine, different fuel.
Different fuel, different enzymes. Prominent examples include renilla luciferase, a macroleuk, from the sea pansy Renilla reformis.
Sea pansy.
Okay, and nanoleuk, which is a more recent development, heavily engineered. Rawleuk has some advantages. It's smaller than Flex, about 34 kilodarios compared to Fluke's 62 kiloday.
Smaller is better sometimes.
It can be beneficial when you need to attach the luciferase to other proteins without creating too large a fusion molecule, or when delivering the gene using smaller viral vectors, for example.
And raw Luke is naturally paired with something else in the sea pansy, right? Leading to a key technique.
Yes, in the sea pansy, the blue light produced by Rawleuk is naturally transferred to a nearby green fluorescent protein, GFP.
GFP. The jellyfish protein.
The famous one. It excites the GFP, which then emits green light. This is a natural process called bioluminescence resonance energy transfer, or BRET.
Bretis, okay.
Scientists have adapted this by fusing Rawleuk to one protein and a fluorescent protein like GFP to another if the two proteins interact or come close together.
The energy jumps from Rawleuk to GFP.
Precisely. The energy from the Rawleuk reaction is transferred to the GFP, resulting in light emission at the GFP wavelength.
So, BRET lets you use light to detect if two specific molecules are interacting or in close proximity inside a cell.
Exactly. It's a powerful tool for studying protein interactions, receptor binding, and other molecular dynamics in living cells. You see light, you know they're close.
Very cool. And then there's nanoleuk. This sounds newer, more engineered.
Nanoleuk is a highly optimized luciferase system developed by Promega. It's actually a significantly engineered version of a luciferase found in a deep-sea shrimp, Aplophorus gracilarostris.
A shrimp luciferase.
Yep, and they really tweaked it. It's even smaller than Rawleuk, just 19 kiloday, making it excellent for fusions. Promega engineered it to work with a specific synthetic substrate called furamazine.
Furamazine. Synthetic fuel for the engineered enzyme.
Right.
How does nanoleuk compare to firefly? Is it brighter?
Yes. Nanoleuk is renowned for its extremely high photon output. It's much, much brighter per molecule than wild-type firefly luciferase. This makes assays using nanoleuk incredibly sensitive.
Brighter means you can detect smaller amounts or see things more easily.
Exactly. Another key feature is that its light emission is ATP independent, unlike the firefly system.
So its brightness doesn't depend on the cell's energy levels.
Correct. This could be an advantage if you want your signal to be independent of the cell's energy status. Makes the measurement maybe simpler in some contexts.
There's always a but, isn't there?
Yeah, often. Like many coentyrazine-based systems, its major drawback for in vivo imaging is its emission wavelength. It peaks at a very blue 456 nanometers.
Oh, back to the blue light problem, bad for tissue penetration.
Exactly. So standard nanoleuk with furamazine is fantastic for bright, sensitive assays in test tubes or cell cultures, but not suitable for seeing deep inside a living animal due to tissue absorption.
And this is where the engineering really shines, right? Scientists aren't just observing the natural systems, they're actively modifying and creating new ones to overcome these limitations. This is where it gets really interesting.
Absolutely. The entire field of engineering bioluminescence is driven by the need to improve upon what nature provides for specific applications.
What are they trying to improve?
Things like trying to increase quantum yield even further,
Yeah.
shifting the emission wavelength to redder light for better tissue penetration,
Redshift, yeah.
improving the stability of the components, making them less toxic, and enhancing their bioavailability so they can get to where they need to go in the body, basically making them better tools.
So how do they go about engineering these systems? What kind of techniques are we talking about?
It's a multidisciplinary effort, really. You've got synthetic chemistry to design and synthesize new luciferin molecules.
Making new fuels.
Right. And then you've got protein engineering techniques like site-directed mutagenesis, making specific changes to the enzyme's DNA, or random mutagenesis.
Just trying lots of random changes.
Sort of, yeah. Introducing random changes and then screening thousands or millions of variants for the ones that have the desired properties, like brighter light or redder light, altering the luciferase enzymes themselves.
Let's focus on that wavelength shift. You mentioned red is key for imaging in vivo. How are they trying to achieve that?
The sources really emphasize this. Getting light emission into the far red 600 millimeter or even near infrared 700 millimeter window is crucial for imaging through blood and tissue, where absorption is minimal.
The tissue optics window.
Exactly. While D-luciferin's peak at 558 millimeter is already better than blue systems like Nanoluke's native form, it's still not ideal for deep targets.
And the sources showed a whole array of synthetic D-luciferin analogs, essentially tweaked versions of the molecule, like a chemical library.
Yes, looking at the images and notes provided, you see chemists systematically modifying the D-luciferin structure to shift the emission wavelength. It's quite impressive.
What are some examples?
There are examples like BTLH2 at 523 millimeter, luciferin pushing into orange at 594 millimeter, Cybluke and Cycluke 1 around 600 millimeters, 5-Bruluke at 625 millimeter,
Getting redder.
and later generation molecules like Bioluminescence pushing past 670 millimeters, and experimental ones reaching towards 750 millimeter, like #40 and #41 in the notes. These are deep red, near infrared.
And little icons next to them in the sources showed they're testing these.
Yeah, they show the progress tested in vitro, in a test tube, then in cellulo, in bacterial or mammalian cells, and crucially, many now tested in vivo in mouse models.
So they're not just making them redder on paper, they're successfully testing them in live animals.
Exactly. Bioluminescence even shows testing in monkeys, indicating potential translational progress moving towards human applications, perhaps.
Wow.
The sources mention ILHT, analog #39 in the notes, specifically being tested in mice, demonstrating its potential for in vivo applications at its 720 millimeter wavelength when paired with the right engineered enzyme.
Right, because you need the enzyme to work with the new fuel.
Precisely. The enzyme's structure determines how it binds the luciferin and how the reaction proceeds, which influences efficiency and wavelength. So scientists are engineering luciferases to work better with these new synthetic luciferins, or to modify the reaction pathway itself to shift the light emission.
How do they engineer the enzyme?
The sources give several examples. Miller, for instance, engineered firefly luciferase mutants. What's interesting is he designed these mutants to be less efficient with the natural substrate, D-luciferin.
Less efficient. Why would you do that? Seems backwards.
It's about creating orthogonal pairs.
Orthogonal, like working independently.
Exactly. Meaning a specific engineered enzyme only really lights up with a specific synthetic luciferin, and not much with a natural one, or with other synthetic ones. They engineered the enzyme's active site to have a higher QIAM for the natural D-luciferin and ATP.
Okay, Curie again. That's enzyme affinity. High Curie means low affinity, needs more substrate. Low Curie means high affinity.
You got it. So they made the enzyme picky, preferring the synthetic substrate. They lowered the enzyme's liking for the natural substrates while increasing its liking for the synthetic ones.
Why is that useful?
This allowed them to use different engineered enzyme-synthetic luciferin pairs simultaneously in the same mouse brain and get distinct signals from each pair.
So you can track two different things at once in the same animal.
Exactly. Enabling multiplexed imaging, tracking multiple biological events at once using different colors or signals. Very powerful.
That's a clever way to use enzyme kinetics for complex imaging. What other engineering approaches are highlighted?
The pressure group took a high-throughput approach. They screened a huge library of 159 Nanoluke mutants against 12 different synthetic luciferins derived from coelentrazine.
Wow, 159 mutants times 12 luciferins. That's a lot of combinations.
It is. Their goal was also to find orthogonal pairs, specific Nanoluke mutants that work optimally with specific synthetic coelentrazines. This massive screening effort identified several promising pairs.
And did they work in vivo?
Yes, they successfully used them for in vivo imaging studies of mammary carcinoma in mice, so that brute force screening paid off.
It's a systematic search through many combinations to find the best partners. What else?
Other examples include Branchini et al., developing a red-emitting chimeric firefly luciferase. This enzyme was engineered to be very efficient, having low count-uppers for both ATP and D-luciferin.
So it works well even if there isn't much ATP around.
It's exactly. Particularly useful for imaging in cellular environments where ATP levels might be limiting, as the enzyme will still function efficiently.
Makes sense.
Pinton et al. Focus on cellular localization. They engineered chimeric luciferases that contain specific targeting sequences.
Like address labels for different parts of the cell?
Kind of, yeah. Directing the light production to discrete compartments within the cell, like the mitochondria or the plasma membrane.
So you can measure ATP specifically in the mitochondria, not just the overall cell.
Exactly. You can get spatially resolved information about cellular energy status. Very detailed.
Amazing precision.
Viviani and coworkers developed a blue-shifted beetle luciferase.
Blue shifted. But we want red.
Hey! Yeah. Seems counterintuitive for in vivo imaging, but this specific enzyme set a record for the lowest parameter for ATP among beetle luciferases, combined with high catalytic efficiency and thermal stability.
So it's super sensitive to ATP.
Extremely. Excellent for highly sensitive ATP detection and could be engineered further for other sensing applications like metal ion or pH changes using radiometric approaches perhaps.
Okay, so different tools for different jobs.
Exactly. And going back to NanoLuc, researchers like AI et al. Created mutants like Toluc and Lumaluc that perform better with synthetic coenantazine analogs like DTZ and APY-DTZ, offering enhanced brightness or better solubility. Though,
Another but.
the source is caution. Autooxidation remains a challenge for many coenantazine analogs, limiting their stability. They can basically react with oxygen on their own without the enzyme, which complicates things.
So chemists and protein engineers are working hand in hand. It sounds like scientists are throwing every tool they have at this, modifying the light molecule, modifying the enzyme, directing them to specific locations. What about using whole bacterial systems as tools? We mentioned them briefly.
Right, bacterial bioluminescence systems, particularly the set of genes called the luxoperon that encodes the enzymes needed for light production, are powerful tools for biosensing.
Biosensing, detecting specific things.
Yeah, you can engineer bacteria to express these lux genes in a couple of ways for different sensing applications. One is constitutive expression.
Meaning they're always on.
Right. The bacteria are always trying to make light. In this case, the total light output from a culture is proportional to the number of living cells. If you introduce a toxin, the cells die, their light output drops.
Ah, so light acts as an indicator of cell viability and toxicity. Fewer lights mean something bad happened.
Exactly. A quick toxicity test. The other approach is inducible expression.
So they only turn on under certain conditions.
Right. Here, the lux genes are placed under the control of a regulatory switch called a promoter that is only turned on in the presence of a specific target compound.
Like what kind of compound?
Could be a pollutant, a metal ion, a specific organic molecule. So when the target compound is present in the environment, the bacteria start glowing, and the intensity of the light tells you how much of the compound is there.
That sounds very useful for environmental monitoring. Testing water samples, maybe?
It is. The sources mention examples of using bacterial biosensors to detect specific analytes like zinc, toluene, and uranium, as well as a range of other chemicals relevant to ecotoxicology. Basically, using glowing bacteria to detect pollutants or specific chemical signatures in samples.
Any drawbacks?
They do note that sample pretreatment is often required to remove substances that might interfere with the bacteria or the light reaction. You can't just dip them in any old pond water usually.
Makes sense. Okay, let's talk about another clever chemical strategy mentioned. Caged luciferins. The name itself makes me think of trapping light somehow.
That's a good way to think about it.
Yeah.
In caged luciferins, a crucial part of the luciferin molecule that's needed for the light-producing reaction is chemically masked or caged.
Hidden away.
Exactly. This mask prevents the luciferin from reacting and producing light. The key is that this mask can only be removed by the specific biological molecule or event you want to detect.
So the light is off until the target is present, and then the target unlocks the cage and turns the light on, like a switch flipped by the target.
Exactly. They function as turn-on probes. When your target may be a specific enzyme's activity or the presence of a particular small molecule is present, it triggers a chemical reaction that cleaves or removes the masking group from the caged luciferin.
And then a light.
The now uncaged luciferin is free to react with its luciferase partner, which is also present, and produce light. D-luciferin is favored for making these caged probes.
Why D-luciferin?
Primarily because, as the sources note, it's generally more stable than coenantazine analogs, making the caged probes more robust and easier to handle.
What kind of targets can these turn-on probes detect? You mentioned enzymes and small molecules.
A surprising range. The sources list detection of specific enzyme activities, various small molecules crucial in biology like glycans, hydrogen sulfide, H2S, hypochlorous acid, HOCl, carbon monoxide, CO, and hydrogen peroxide, H2O2.
Wow, those are all important signaling molecules or indicators of cell stress.
They are, and even metal ions like copper, iron, and cobalt can be detected this way.
That's incredibly versatile. And what's the advantage of using a bioluminescent turn-on probe compared to a traditional fluorescent one, which also glows when a target is present?
The major advantage is that you don't need any external light source to excite it.
Ah, no laser needed.
Right. Fluorescent probes require you to shine light on them to make them glow. And that excitation light can cause background noise, interfere with the signal, or even damage sensitive biological samples, phototoxicity.
Right. Background fluorescence can be a problem.
With a bioluminescent probe, the light is generated internally by the chemical reaction itself, triggered only by the presence of your target. This results in a much higher signal-to-noise ratio, meaning cleaner, more sensitive detection. No background from excitation light.
Any downsides to caged probes?
The sources do mention a potential trade-off. Caged bioluminescent probes might sometimes need to be used at higher concentrations in cell assays compared to fluorescent ones, which could potentially subtly impact cell biology, though this depends on the specific probe, something to be mindful of.
But they're successfully using these caged probes in vivo, in actual animals.
Yes, several caged luciferins have been used for imaging specific targets inside living mice, demonstrating the power of this approach in a physiological context.
And the sources highlight a particularly elegant recent development, something about mitochondria.
Yes, a caged luciferin probe designed specifically to measure changes in mitochondrial and plasma membrane potential in vivo.
Mitochondrial membrane potential, that's getting very specific inside the cell. What does that tell you?
It's a key indicator of mitochondrial health and function. Mitochondria are the cell's powerhouses, and that membrane potential is crucial for making ATP. This probe is called a mitochondria activatable luciferin, or ML probe. It's designed to non-invasively monitor the health and function of mitochondria by reporting on the electrical potential across their inner membrane, and also the potential across the outer plasma membrane.
How does it work? Sounds complicated.
Uses a very clever mechanism involving a bioorthogonal reaction.
Bioorthogonal? That sounds technical. What does that mean in practice here?
It means a chemical reaction that can occur inside a living system without interfering with all the complex biological processes happening there. It's like doing specific chemistry without messing up the cell's natural chemistry.
Okay. So how does the MAL probe use this?
This ML probe works via a specific bioorthogonal reaction called a Staudinger ligation. The probe molecule is engineered with a Staudinger reactive group, and another molecule called azeto-TPP1 carries the other reactive group, an oxide.
Two parts that react together.
Right, and both the probe and the azeto-TPP1 molecule are also decorated with chemical tags called TPP groups.
TPP groups, what do they do? Like little flags?
TPP stands for triphenylphosphonium. These groups act like shipping labels or GPS tags that specifically direct the molecules to the mitochondria. Because mitochondria have a negative membrane potential that attracts the positively charged TPP groups. So the caged probe and its partner azeto-TPP1 are actively taken up and concentrated in the mitochondria.
So the reacting parts get delivered right to the target organelle.
Exactly. Once inside, the Staudinger ligation reaction between them proceeds. This reaction uncages the luciferin.
And how does this relate to the mitochondrial potential? That's the key bit.
This is the clever part. The rate of this specific Staudinger ligation reaction, which ultimately leads to the caged luciferin being uncaged and becoming active to produce light. That rate is proportional to the mitochondrial membrane potential and also reflects the plasma membrane potential.
Wow. So the electrical state of the mitochondrial membrane influences how fast the uncaging chemical reaction happens, which in turn determines how much light is produced.
Exactly. Higher potential, faster reaction, more light, or vice versa, depending on design. The light output you measure is then a direct, non-invasive report on the mitochondrial and plasma membrane potential inside the living animal.
That's incredible precision in targeting and reporting, linking cell physiology directly to light output.
It's a highly sophisticated way to link a specific cellular physiological state to a bioluminescent signal. Really cutting-edge stuff.
Okay, shifting gears slightly. What about split luciferases? They sound like the enzyme is in pieces.
That's precisely the concept. Split luciferases are powerful tools primarily used for studying interactions between molecules, most often proteins.
How do they work?
The luciferase enzyme is genetically engineered into two separate fragments, typically the N-terminal, start, and C-terminal end parts. Individually, these fragments are inactive. They can't catalyze the light reaction on their own.
So you attach one piece to protein A and the other piece to protein B that you think might interact.
Yes, you genetically engineer your cells so that one piece of the split luciferase is fused to protein A and the other piece is fused to protein B, the two proteins you're interested in studying.
And then what happens?
If and when protein A and protein B interact or come into close proximity within the cell, They bring the two inactive pieces of the split luciferase together.
And when the pieces are close, they reassemble or interact to form a functional enzyme. Click back together.
Exactly. When the two protein partners bring the split luciferase fragments close enough, the fragments can reassociate or complement each other to form a functional active luciferase enzyme.
And then you add the fuel.
Then when you add the luciferin substrate, the reconstituted enzyme catalyzes the light-producing reaction. So light output becomes a direct signal that protein A and protein B are interacting. No interaction, no light. Interaction, light.
That's a powerful way to visually detect molecular interactions in real time. What are some examples from the sources? Where is this used?
The sources mention using split nanolute, because it's bright and small, good for fusions, to study protein interactions, like the interaction between plant receptor kinases FLS2 and BAK1.
Plant biology, too.
Yep, showing how a bacterial peptide triggers this interaction in plant cells. Split luciferase assays are also being applied to study viral assembly or processes.
How viruses put themselves together. Right?
Exactly. Or how viral proteins interact with host cell proteins, and also to detect protein aggregation in human cells.
Protein aggregation, like in Alzheimer's or Parkinson's.
Precisely. Relevant to neurodegenerative diseases where proteins misfold and stick together, the split luciferase system can report on that aggregation process happening inside cells.
So you could potentially use this to screen for drugs that prevent proteins from aggregating, find compounds that make the light go down.
Absolutely. If a drug prevents the protein interaction or aggregation, the luciferase fragments don't come together, and you see less or no light. This leads us directly to another major application of bioluminescence, high-throughput screening, or HTS.
This is huge in drug discovery, right? Testing vast numbers of potential medicines very quickly.
Exactly. HTS is designed to quickly test thousands or even millions of compounds to find those that have a desired effect on a biological target. And bioluminescence is perfectly suited for HTS.
Why is it so good for that?
Because it provides a very fast, sensitive, and quantifiable light signal that's easily measured automatically in multi-well plates. You can test many compounds side by side very rapidly.
What's the general setup?
The general approach is to create a biological system, a cell line, a pathogen, a purified protein complex where a biological process of interest is linked to bioluminescence.
How is it linked? How do you connect the biology to the light?
Often, the gene for a luciferase, like Fluke, Arluc, or Nanoluc, is introduced into the system. Its expression or activity is made dependent on the biological process you want to study.
Can you give an example?
Sure. If you're looking for drugs that block a specific gene's activity, you might put the luciferase gene under the control of that target gene's promoter. If the drug inhibits the target gene, luciferase expression drops, and you see less light.
Okay, light level reflects gene activity.
Or if you're looking for drugs that kill a pathogen, you might use a pathogen engineered to constitutively express luciferase.
Always glowing, unless it's dead.
Right. Drugs that kill the pathogen reduce cell numbers and thus reduce light output. The amount of light tells you how many pathogens survive the drug treatment.
And the sources give an example related to viruses, something recent.
Yes, they mention an assay developed to screen for inhibitors of coronaviruses using a viral strain that was engineered to express Raulic.
So they made the virus glow.
Essentially, yes. By measuring the light output, researchers could quickly assess how well different compounds inhibited viral replication. Highlight meant the virus was replicating well and the drug wasn't working. Low light meant the drug was inhibiting the virus.
A fast way to find potential antiviral drugs.
Exactly. Bioluminescence provides that clear, rapid readout needed for the massive scale of HTS.
It's clear bioluminescence has transitioned from a biological curiosity, fireflies and glowing fish, to an indispensable tool powering fundamental research, drug discovery, diagnostics, and environmental monitoring.
It truly has. And the progress is accelerating, particularly with the engineering efforts we discussed. The ability to tweak and improve these systems is huge.
And the sources summarize this trajectory in imaging applications. Going deeper.
Yeah, starting from relatively simple imaging, just under the skin, subcutaneous, to now being able to tackle challenging targets deep inside the body, like tumors in internal organs or processes within the brain.
Things you couldn't see before with bioluminescence?
Not easily, or not clearly. But now it's possible thanks to the development of those brighter, more stable, and crucially, red-shifted engineered systems we talked about.
And this often follows a path from cell culture to mouse models and then potentially towards clinical applications. That's the usual pipeline.
That's the standard progression, yes. New systems are first validated in simple cell cultures, then tested in small animal models like mice to assess their performance in vivo, including delivery, bioavailability, toxicity, and imaging depth, before potentially moving towards larger animal models or eventual therapeutic or diagnostic uses in humans.
So, incredible progress, but what are the remaining hurdles? What are the limitations holding this field back? It can't all be solved yet.
No, definitely not. The sources are clear that despite the advancements, there are still significant challenges. One is quantum yield.
Still trying to get more light out for the energy put in.
Exactly. Even the best natural systems like Firefly D luciferin at 41% max, or engineered ones like Nanoluke, though intrinsically brighter, the overall system efficiency can vary depending on substrate stability and delivery, still fall short of the theoretical maximum for converting chemical energy to light. There's still room to make them inherently more efficient light emitters.
And the color problem is still there for some systems, right? Especially the really bright ones.
Yes. While there's been great progress in engineering redshifted D-luciferin analogs and pairing them with engineered fluke variants, getting those beneficial redshifted nanoluke variants, which promise extreme brightness and good tissue penetration, commercially available and widely adopted, is still somewhat a work in progress.
What about the molecules themselves, the luciferins?
For many synthetic luciferins, issues remain with stability in biological solutions, compatibility with cells, cytotoxicity at necessary concentrations, and bioavailability.
Making sure they get where they need to go in the body without causing problems.
Right. Ensuring the molecule can reach its intended target in the body after administration and not break down too quickly or harm the cells. And coenzyme analogs, while often enabling brighter systems like Nanoluke, are still prone to that auto-oxidation problem we mentioned, limiting their utility in some applications that require long-term stability.
So it's not a solved problem. Developing better lucifer and luciferase pairs is still a very active area of research. Lots of chemists and biologists still busy.
Absolutely. Scientists are constantly searching for and engineering new combinations with improved properties, better light, better stability, better targeting.
And maybe finding totally new systems in nature.
That too. There's also the exciting prospect of exploring and cloning luciferase systems from less studied organisms, like the autoluminescent fungi we briefly mentioned. These organisms have evolved unique ways to produce light, which might yield entirely new classes of luciferases or luciferins with novel properties beneficial for biotechnology. Nature might still hold some surprises.
Speaking of fungi and the more aesthetic side of bioluminescence, it's not just scientists who are captivated by this natural light show. It also inspires artists.
It certainly does. The sheer visual impact of living light has opened up unique avenues for creative expression. It's just inherently cool to look at.
What kind of art are we talking about?
The sources mention examples of living art created using bioluminescent organisms like bacteria and single-celled marine dinoflagellates.
Like the famous bacterial art on petri dishes, painting with glowing microbes.
Yes, exactly. Where artists use colonies of glowing bacteria as their medium on agar plates. The piece isn't static either.
How so?
It changes over time as the bacterial colonies grow, interact, and eventually die off, causing the light to wax and wane. It's a dynamic, living canvas that also serves as a powerful way to engage people with microbiology.
That's amazing. Any specific artists mentioned?
Artists like Yvonne Koo and Hunter Cole are highlighted for working with or being inspired by bioluminescence in their pieces.
It's such a direct fusion of science and art. And our sources even suggest that you, the listener, can actually experience this living light firsthand, not just look at pictures.
You can. The sources include practical notes on how to collect bioluminescent bacteria from natural sources and culture them yourself. A bit of DIY bio.
From seawater or even seafood, like glowing fish from the market. Is that real?
Yes. As we mentioned, bioluminescent bacteria are common in marine environments and can rapidly colonize dead marine organisms. So you can collect samples from fresh seawater or from the surface of fresh marine fish, shrimp, or squid bought from market.
And then grow them. How?
Using a simple growth medium, often referred to as SWC agar, basically seawater with some nutrients like yeast extract, peptone, and glycerol, you can streak these samples onto petri dishes. If luminous bacteria are present, colonies will grow and start emitting light, usually best seen in complete darkness after a day or two.
That sounds like a fascinating, accessible project for anyone interested in a little DIY biology. The sources detail the basic ingredients and process.
They do, and importantly, they include basic safety precautions. While the common bioluminescent bacteria you're likely to isolate this way, primarily Vibrio or Photobacterium species, are generally not considered pathogenic to healthy humans.
Still handle with care, right?
Absolutely. Common sense dictates treating any microbial culture with respect, washing hands thoroughly, keeping the work area clean, and avoiding direct contact with the cultures. Standard microbiology practice.
And do they glow forever?
The notes also mention that fresh cultures glow brightest. The luminescence might fade after a few days as nutrients are depleted, but you can keep the glow going by transferring a bit of the culture to fresh media, subculturing.
That's a really cool way to engage directly with this phenomenon. And that hands-on idea connects to another fascinating thought from the sources, specifically in notes from a Reddit discussion about bioluminescent fungi, the idea of trying to selectively breed them for a brighter glow.
Yes, that was an interesting thread, exploring the practicalities and potential of taking that natural glow and trying to enhance it through breeding. Someone asked if it was possible to selectively breed bioluminescent fungi to make them brighter, like cultivating plants for a desired trait.
And is that feasible? Could you make brighter glowing mushrooms just by picking the best ones over generations?
The general consensus in the discussion, drawing on principles of genetics and cultivation, was yes, selective breeding is definitely possible for organisms like fungi.
Why fungi?
They have relatively fast life cycles compared to, say, trees, allowing you to go through multiple generations within a reasonable time frame. The basic process would involve growing a population of bioluminescent fungi, perhaps from spores of a naturally glowing species,
Okay.
measuring the intensity of the light they produce using something like a sensitive light meter, or even just by eye, identifying the individuals or colonies that glow brightest, and then using the spores from those individuals to start the next generation.
So you'd literally be picking the glowiest mushrooms and breeding them. Sounds simple enough.
Effectively, yes. Repeating this process over many generations should, in theory, lead to a population with a heritably brighter phenotype. But
The but again.
participants in the thread pointed out that success would depend heavily on controlling environmental factors, temperature, humidity, the specific substrate or food source they grow on.
Because those affect the glow too.
Exactly. These can significantly influence how much light a fungus produces, independent of its genetics. They also discussed potential trade-offs.
Trade-offs. Like what?
If the fungus is putting a lot of energy into making light, that energy isn't available for growth, reproduction, or defense. So aggressively breeding for brightness might, in some conditions, lead to strains that are less hardy or grow more slowly.
That makes perfect sense. Nature always balances costs and benefits. You can't get something for nothing.
Right, a classic example of evolutionary trade-offs. The discussion also briefly mentioned genetic engineering, like using tools such as CRISPR as another way to enhance luminescence.
A more direct approach.
Potentially, noting that while kits exist for organisms like yeast, applying this directly to bioluminescent fungi might be more complex currently. Still early days for widespread fungal genetic engineering kits.
So maybe breeding is more accessible for now.
For the hobbyist,
Right.
probably. But the overall sentiment of that thread, and indeed the broader field, is one of pushing boundaries, exploring potential, and trying to understand and harness this amazing natural phenomenon. It's a great example of how bioluminescence sparks both rigorous scientific investigation and imaginative curiosity, even inspiring potential DIY bioart and cultivation projects.
We've journeyed from the fundamental chemical reaction that creates light in living things, explored the incredible diversity of ways organisms use this light for survival and interaction in nature,
From camouflage to communication.
delved deep into how scientists are engineering these systems into powerful tools for research, diagnostics, and screening, and even touched on how this natural magic inspires art and citizen science.
Bioluminescence truly is a remarkable meeting point of chemistry, biology, and engineering. It's a fundamental aspect of life on Earth and a rapidly growing frontier in biotechnology.
It's more than just pretty lights.
Much more. The ability of organisms to produce their own light is not just beautiful, it's proving to be profoundly useful.
As researchers continue to engineer ever brighter, more stable, and more precisely targeted bioluminescent systems moving toward faster assays, deeper imaging, and more sensitive detection, how might these natural flashlights fundamentally change how we understand disease, monitor our environment for subtle changes, or even imagine illuminating aspects of our future we can't yet conceive? It raises an important question.